Hello everyone, my name is Ainur Jumatayevna. I'm a biology teacher of Nazarbayev Intellectual School in Simi. And today I present 11th grade curriculum, Term 3, Unit 11.3, a Molecular Biology. Topic, DNA replication and genetic code. Today we discuss a two lesson objective. There are describe the process of DNA replication, and last one, it's explain the properties of genetic code. So let us start our lesson from brainstorm questions, such as, what is DNA replication? Take a time to think about that. The first question is like that. Where does it take place? And the second question, when does it take place? So these questions help to you to remember previous lessons, which are necessary for today's lesson. Let's answer the previous questions. DNA replication is a biological process that occurs in a living organism and copies their DNA. Take a note. It's necessary to know that DNA replication takes place during the C phase. Also, we say it's synthesis phase, in interphase, cell cycle. If you remember, cell cycle delete in two major groups, like interphase, which take a big time of cell cycle, and mitosis, which take a short time. Also, interphase has three phases, like GAP1 phase, synthesis of DNA replication, and GAP2 phase. What about mitosis? It's divided in four phases: prophase, metaphase, anaphase, and telophase. Propose DNA replication models. There were three hypotheses suggested to explain the process of DNA replication. One of them is known as semi-conservative replication. The other hypotheses are known as conservative replication and dispersive replication. So right now we discuss about semi-conservative replication. DNA replication would create two molecules, each of which would be complex of an old, parental and daughter strand. So here you see a diagram. The first DNA, its name is original parent strand. In other words, we say it's parental strand. So it's separated in two groups, two DNA helix. One of them will be parental strand and second of them will be daughter strand. And after that, this is separate itself in two groups. So you see this. It means second generation daughter strands. Let's talk about conservative replication. So conservative replication, the DNA replication process would create a brand new DNA double helix made of two daughter strands, while the parental change would stay together. So here you see a diagram conservative replication. And one of them, the first, it's parental strand, or we say it original parent strand. It starts to separate itself into DNA double helix, and one of them will be daughter strand, and second DNA double helix will be parental strand. After that, the daughter strand and parental strand divide itself and give to us DNA double helix. So let's see this part. It means after second replication cycle. And last one is a dispersive replication. Dispersive replication is a replication process which creates two DNA double chain, each of them which parts of both parent and daughter molecules. So here you see a diagram dispersive replication. So the first is original parent strand. Like I said before, it's separated in two double helix DNA. And shorter segments, which will be parental strand, and one shorter segment, it will be daughter strand. So after that, it delete or separate itself. And give to us dispersive replication. We stop in a history of semi-conservative replication. The method of DNA replication proposed by Watson and Crick is known as semi-conservative replication. Since each new double helix retains one of the two strands of the original DNA double helix. The evidence for these mechanisms was provided by a series of classic experiments carried out by Mendelssohn and Stachel in 1958. Its take experiment. SLA has a single circular chromosome, and when cultured of these cells were grown for many generations in a medium containing the heavy isotope of nitrogen, 50N, all the DNA labeled with 50N. The cell containing DNA labeled with 50N were transferred to a culture medium containing the normal isotope of a nitrogen and allowed to grow. The cells were then left for periods of time corresponding to the generation time for SLA. That is the time needed for the cells to divide once and therefore the DNA to replication once. Samples were removed and the DNA extracted in centrifuge at 40,000 times, graphing for 20 hours in a solution of cesium chloride. During centrifuge, the heavy cesium chloride molecules began to sediment to the bottom of the centrifuge tubes, producing an increasing density gradient from the top of the tube to the bottom. The DNA settles out where its density equals that of the cesium chloride solution. When examined under ultraviolet light, DNA appeared in the centrifuge tube as a narrow band. The position of the bands of DNA extracted from cells grown in 50N and 40N culture media. So this is a history about SLA, how it was cultured. So let's speak about basic concept of DNA replication. Right now you will see a diagram about DNA replication. The first it will be parental strand. So it's old strand. The next one is after separating. So here we use a helicase. Each parental strand become a template. And then next you see here, it's a new strand with a mother strand. So it's a daughter strand with a mother strand. So in short we say it is complementary pairing. And here in the end we see each daughter molecule has one parental strand and one new strand. DNA replication in higher organisms. You see diagram with DNA helix, which start to replicate. And the strand of DNA have, the first it will be 5 prime, and the opposite of that is second, has 3 prime. Before we start about DNA replication, you need to take a note. During DNA replication, the 5 prime end of a free nucleotide is always added to the 3 prime end of an exiting DNA strand. The strands are antiparallel. So here you see DNA structure. We talked about in previous lesson. You know that DNA structure has phosphodesoxyribones and 4 types of nucleotides. So they are thymine, adenine, guanine and cytosine. Also, we talked about in previous lesson about Chargaff rule. So, the Chargaff is a young science which suggests about that. And according to the Chargaff rule, you know that adenine always complementary binding with thymine, and their binding with hydrogen. They have 2 hydrogen binding. What about cytosine? It's always complementary binding with guanine, and their complementary binding is 3 hydrogen bound. So adenine and guanine is purines. What about thymine and cytosine? It's pyrimidines. These strand complementary binding nucleotides can separate using unwinding enzymes, or in other words, they said helicase enzyme. Let's talk in more detail. First, strand separate using unwinding enzyme. So here you see unwinding enzymes. In other words, we said it's helicase. So helicase can separate template strand and give it. Helicase move along the DNA separating and unwinding the 2 DNA strands by breaking the hydrogen bonds between complementary bases. DNA helicase completes the splitting of the strand, and as it is doing this, free nucleotides start to be attached to their complementary base on the templates. So here you see a diagram how DNA helicase start to separate. So one of them is origin of replication. And here it will give replication fork. The 2 strands are split, unzipped, forming the replication fork. So here you see a diagram about replication fork. So it has parental strand and daughter strand. When it unzips, it starts to become template strand. Or in other words, we say it's old parental strand. So here you are. They can't attach each other because they have single strand binding proteins. So single strand binding proteins couldn't allow to attach each other. Let's see in more details. Here you see a helicase and SSB proteins. So SSB proteins binds on the separated strands and prevents the strands from coiling. Also, DNA helice helps unwind the DNA head of the replication fork and prevent it from coiling. Here you see DNA polymerase. Let's draw in other illustration. The first what will be on the template strand, it's RNA strand. Or we say it's RNA primase. After that, we take DNA polymerase. It gives DNA strand. Once the strands are exposed, RNA primase lays down a primer, allowing DNA polymerase III adding nucleotides. So, when strand unwind, free nucleotides tied to binding complementary. So here you see DNA separating. And right now, you see 3 nucleotides. So you know it's 4 type. So let's just see and zoom it. Here you see 3 types of nucleotides and 1 type. Let's do imagine that it's adenine triphosphate. So it's adenine. And when adenine attached to the thymine, it needs a ATP. So here you can take a note. Also, a free nucleotide is attached to its complementary base. On the template, it cannot join until it is attached by energy. This energy is supplied by ATP. Here you see how free nucleotides binding DNA polymerase. So it's link the nucleotides. Once the nucleotides are in position, phosphodiester bonds are formed between their phosphate and desoxyribose sugars. This is done by the enzyme DNA polymerase. So, in the first diagram you see DNA replication. Let's zoom it. And here you see DNA polymerase, also RNA primase, helicase, and 2 different types of strands. So we need to describe it in more details. And let's see that. DNA polymerase adds nucleotides on the strand that runs 5' to 3', adding nucleotides on the 3' end. Here you see direction of replication. So take a note that DNA replication can only add nucleotides to the 3' end of the DNA. This causes the new strand to build in a 5' to 3' direction. In the beginning of new strands occur RNA primer. Adds start sequence of RNA to both primer and allows the DNA polymerase III to bind and start replication. DNA polymerase III adds nucleotides to each template strand in a 5' direction 3', because the nucleotides can be added only at the 3' and it gives direction of replication. Here you have 2 types of strands. The first is leading strand. Is made in the 5' to the 3' direction by continuous polymerase. So we say it continuously polymerization as the 3' growing tip. So this is illustration of leading strand. Strand of DNA being replicated continuously. Here you see leading strand. Second strand is lagging strand. The lagging strand is synthesis discontinuously against overall direction of replication. This strand is made in many short segments. It's replicated from the replication fork towards the original. Here you see leading strand. Series of short segments on the leading strand. It's Okazaki fragment. It is a science. So here on one part of short segments, you see Okazaki fragment. And next part will be Okazaki fragment. Let's increase it. So this is Okazaki fragment. You see it's in a big illustration. Okazaki fragments joins together by an enzyme's ligase to make one strand. So here it's joining Okazaki fragments, and it's joining via the enzymes like DNA ligase. And let's draw it in big illustration. So this is DNA ligase. DNA polymerase initially makes about 1 in 10,000 base pairing errors. So here you see a different kind of DNA replication. And you see one base is error. So the question like that: how does new DNA correct their mistakes? Is it possible? Here you see a little piece of DNA replication. And also DNA polymerase, which added nucleotides. For instance, it's thymine. So thymine complementary binding with adenine, as we taught before. So the next nucleotides on replication, you see adenine, but you see adenine can't bind with cytosine. So the question is like that. And this question we need to find. So what will do DNA polymerase? In the beginning, DNA polymerase sees the errors. Enzymes proofread and correct these mistakes. In this picture, you see proofreading. So adenine go away and lose his energy. And new nucleotides will come. So it will be guanine, because guanine complementary binding with cytosine. So here you see how cytosine binding with guanine. It means extension. The new error rate for DNA that has been proofread is 1 in 1 billion base pairing errors. DNA polymerase proofreads, deletes the wrong nucleotides and resumes synthesis. So let's just sum up our lesson. And the first what you need to know is like that. The enzyme DNA helicase breaks the hydrogen bonds between the base of two strands. Second, the double helix separates into its two strands. Thirds. Each exposed strand now acts like a template strand. Fourths. Complementary free nucleotides are attached to the exposed base on the strands. Fifths. Energy, ATP, is required to activate the free nucleotides and attach to base. 6. Once the nucleotides are in place, their backbone is joined together by DNA polymerase. 7. Two new DNA strands are produced. The leading strand is called continuously replication, while the lagging strand is discontinuously replication. Next, the lagging strand is made in many short sequences, so it's a Okazaki fragment. The segments joined together by an enzymes like DNA ligase. Each of DNA strand contains half of the original strand and half of the new strand. Let's decide the task which has a student. And right now, he's thinking about that. So he has two big questions. Let's look to his task. So in the task, you see DNA, one of the strands. It goes from 5' direction to the 3'. And it have nucleotides like cytosine, guanine, thymine, adenine, thymine, and guanine. And question sounds like that. What would be the complementary DNA strand for the following DNA sequence? So, let's remember our previous lesson. If you remember that the strands is antiparallels. So here you see a diagram how the strands located opposite each other. So next DNA will be antiparallel. It's go from 3' direction to 5'. Next rules, you must know about Chargaff's rule. You know that the number of adenines is equal to the number of thymines. The number of cytosines is equal to the number of guanines. So here we are writing guanine, cytosine, adenine, thymine, adenine, and cytosine. Next rule. Sense strand runs from 5' to 3'. So it's sense, it's coding strand. Next strand, it will be template strand. So it runs from 3' to 5'. So next DNA is template strand, or we said antisense strand. Uses the template or basis for making, it's mRNA, messenger RNA. So the question is like that. What is the mRNA strand that would be copied from this template strand? So this is mRNA. It's going 5' direction to 3'. And you know that in mRNA, thymine change to the uracil. So it's look like that. Cytosine, guanine, uracil, adenine, uracil, and guanine. Let's decide next task using previous decision from the task. So you know that DNA one strand and DNA next strand start from nucleotides. So this strand is coding and next strand is template strand. And from template strand we take mRNA. And also we found mRNA nucleotides. So here you see what's the common between this task and genetic code. So you see one of the part of genetic chart, one of the type. And we need to decide this question. The genetic codes is a set of rules by which information encoded in genetic materials, DNA or RNA sequence, is translated into proteins amino acid sequence by living cells. This is, as you see, DNA double helix. And one of the strands we said coding strand. And next strand, it will be template strand. So we can take template strand and found mRNA. So here you see the first strand is template strand. And next, it will give to us messenger mRNA. So this process name is transcription. It occurs in nucleus. So the genetic code is a set of 3 letters combination of nucleotides called codons, each of which corresponds to a specific amino acids or stop signals. So you see a codons. From 3 nucleotides, we will take 1 codon. So it's first codon. Next 3 nucleotides joins and give to us second codon. And last 3 nucleotides will give to us codons sets. So these codons will give to us a different type of amino acids, like arginine, tyrosine, tryptophan. As you know in previous lessons that amino acids, it's a proteins, yeah? So proteins, it's a polymer, and amino acids, it's monomer. And you know that we have 20 types of amino acids. So this process we say translation at ribosome. So it's happening in cytoplasm. So here we need to decide next task using previous task. So you know about DNA strands, and we found next template strand. So here you see coding strand. From coding strand we found template strand. And after from template strand we found mRNA. So it's go from 5' to 3' direction. And also you found these nucleotides. So the question sounds like that. How can we use a genetic code chart to find amino acids? And we need to help these students. So she's thinking about that. And she needs to find type of amino acids. So AA, it will be capital letter. Amino acids we can take from mRNA. It's like that: cytosine, guanine, uracil, adenine, uracil, and guanine. We will use genetic code chart. So it will be round. From first cycle we take cytosine and mag it. Second, it will be guanine. It's located in second cycle. From third cycle, we need to find uracil. So we mark it, and after that we can find a type of amino acids. It will be arginine. And in notebook, we can write like shorter, like ART. Next, we found A, adenine. So it's located in first cycle. After that, we need to find uracil. It's located in second cycle. The last one, we need to find guanine. It's located in third cycle. And all of them will mark and found the type of amino acids. It will be methionine. We take this methionine and mark it in our book like met. So we found it. Let's decide this task in different way. So you have DNA, coding strand and template strand. Also, we found mRNA. It have cytosine, guanine, uracil, adenine, uracil, and guanine. So we will find this task using other type of genetic code chart to find amino acids. We wrote amino acids with capital letter big A. So we start to find our nucleotides from first letter, second letter, and third letter. The first letter is located cytosine. We mark it and let to find guanine. It's located in second letter. And uracil will be located in third letter. We mark it and join it. And we'll find the type of amino acids. It will be arginine. Also, we need to write in our notebook. So here you see ART. The next amino acids we need to find. So the first it will be adenine. It's located in first letter. In second letter we will find uracil, and in third letter we need to find guanine. So we joined it, and it gives to us the type of amino acids like methionine. So in other words, we said it's start codon, and mark in our copybook.